Dear students, in this video we are going to discuss another very important instrument of the amphibian physiology experimental setup, that is Kymograph or Sherrington's recording drum. As we all know, this is the experimental setup containing two circuits, primary circuit and the secondary circuit, and we have divided the instruments in this circuit in this experimental setup under three headings. Some are stimulating appliances, some are recording appliances, some are miscellaneous. So the Kymograph machine, it comes under the heading of recording appliances. That means when we are giving electrical stimulus to the nerve muscle preparation, so there will be some mechanical changes of the muscle contraction, relaxation, or something else. So that will be recorded by this Kymograph machine. So Kymograph provides a uniformly rotating smooth surface for recording the movement of the lever. It consists of the following parts. What are those parts? Electric motor, gears, clutch, leveling screw, shaft, contact button, projecting striker, and the drum. And if the question comes in this way, identify the instrument and enumerate the parts of it, then you have to say this is Kymograph or Sherrington recording drum, and these are the parts of this instruments. So in this instrument, this, as it is it has a rotating drum, so it has an electric motor which runs on 220 volts. We already know that in the experimental setup, when we give electrical stimulation to the nerve muscle preparation, that is a separate electrical setup. Its source is 6 volt DC. Then when the primary circuit is complete, there will be current in the primary circuit, that is continuous, which will be induced in the secondary circuit, and that induced current is a phasic current that will through the electrode will stimulate the muscle. So in this circuit we can see here, so the secondary circuit we give the phasic or induced current, but for rotation of the Kymograph, you can see another separate plug point is attached here, and this helps in the rotation of the drum of the Kymograph. So that runs in 220 volt. It is separate from the experimental electrical source. So this is about the electric motor. Then come to the gear. At the front portion of the instrument, you can see there are some markings present here. These are employed for changing the shaft speed. For example, for if I want to record the SMT or simple muscle twitch, the speed can be 620 millimeter per second. Okay, if I want to record some other experiment, then I can increase the speed also. Okay, so in this way, there is a button here, so by which we can change the position of the button, and accordingly the speed will be changed. So this is known as the gear. At the back of the instrument we have the clutch. Okay, what is the function of the clutch? Thus, like car, this must be used every time before changing the gear. Okay, and it disengages the gear from the axis of the motor. If we don't make it neutral and change the gear, so there is chances of breaking the axle. So every time you need to change the speed of the gear, you have to use this clutch which is present at the back of this instrument. Then we have the leveling screw. It helps in bringing the base of the Kymograph to a horizontal plane. So these are the leveling screw at the below. Now we come to very important part of this instrument, that is the shaft. This shaft over which actually we place the drum. This is disposed vertically and can be rotated at different speed. And it has a groove on one end. You can see here on the right hand side, and a screw on the top. Okay, so in this groove actually we place the drum and we fix the position of the drum with the help of that screw. A small rectangular plug mounted on the shaft it bats it actually bats out through the groove, and it can be raised or lowered along the groove by rotating the screw at the top. Okay, suppose my myograph board is a bit less lower position. Okay, so I can make the drum down. Okay, if the my experimental nerve muscle preparation, it is a high up, so I can raise it. Okay, even when I want to remove the drum, I can also move it up. Okay, so it can be raised or lowered along the groove by rotating the screw in the top, and it supports the weight of the drum and it prevents it from sliding down. So this is very important, that is the shaft. Another important part of the shaft is, at the lower part of the shaft we have contact button and the striker. Okay, contact button is actually present on the stage of the kymograph on the motor. Okay, so this contact button is specially designed knob mounted on the kymograph. Okay, we can see here on the there is a box around it. So this is the contact button. It is specially designed knob mounted on the kymograph, and the circuit, we know that circuit gets completed only when the striker at the lower end of the shaft, it touches this contact button. Okay, so the circuit gets completed when it gets pressed by the rotating projecting striker, and it gives the us actually the point of stimulation. So what are the parts we have discussed till now? We have discussed about the motor, we have discussed about the gear, the back side, we have the clutch, we have the shaft, and we have the contact button. Then we have the projecting striker. Striker is actually present at the lower end of the shaft. Okay, so what is a striker? So if we all know how to play carom, so in the carom board we also have a striker. So that striker actually strikes. So here this striker, it will strike the contact button and complete the circuit. Okay, so the projecting striker, it is a pair of these. One point I am going to disclose here, these striker can be separated. Actually, they are placed one upon another. Okay, so a pair of this striker is there. It is fixed at the base of the shaft. They can be kept close together. We have seen that. We have thought that it is only a single striker, or it can be drawn apart by a desired distance. Accordingly, on each rotation of the drum, the contact button will be pressed either once or twice in close succession at a precise interval of time. So, if I want to give two successive stimuli, so I will separate the project this striker, and I will adjust the distance between them, so that the second stimulus, if I want it in the latent phase, then it will come in the latent phase. If I want it in the contraction phase, it will come in the contraction phase, and we will see what happens if two successive stimuli it is given to the nerve muscle preparation. Okay, so this is the beauty of this instrument. Very simple experimental setup we have, but in this also we can do so many types of experiment to see the properties of skeletal muscle. So this is the way we can see, we can separate it. The projectors are separated, and when one projector is coming, it is touching the contact button. So there is the circuit is complete, so current is there, and then it is moving. Another striker is coming and it is creating another circuit. So another stimulus is coming and giving the stimulation to nerve muscle preparation. So in this way, you can see that it is moving in this way. Okay, so one point, one stimulation, another point, another stimulation. So now come to the effect of successive stimuli. Okay, so here we want to discuss about these two successive stimuli. Separate the projecting strikers slightly so that they strike the contact button in close succession. Now, how can we adjust the distance? Okay, so for that we have a good calculation for it. For example, in a SMT, you have measured by scale that the latent phase has a duration as a distance of 1 centimeter. Okay, it is not in second in time tracing, but you have placed a scale and measured it. It is 1 centimeter. So first half of the latent phase is 0.5 centimeter, next half is the 0.5 centimeter. Okay, and we know that if we give the second stimulation in the latent phase, then there will be no change, because it will be the muscle is in absolute nerve is in the absolute refractory period. So we know the property, and we have proved this property by these experiments. Now if we want to see this in this experimental setup, how we will adjust it? Okay, so the diameter of this cylinder or drum, it is 6 inches or 15 centimeter. Okay, so its perimeter is 2 pi r. So 2 pi into radius, that is 2 pi 7.5, that is around 47 centimeter. So their perimeter is 47 centimeter. Okay, so now if we rotate it for 360 degree. Okay, if the rotation is 360 degree, what will happen? So one complete rotation is 360 degree, we can say. Okay, so 360 degree, it is traveling 47 centimeter. Okay, so we can say 1 centimeter will be equivalent to what? It will be equivalent to 8 degree. Okay, again I am repeating, the diameter of the cylinder is 15 centimeter, so radius is 7.5, so perimeter is 2 pi r, that is 47 centimeter. One complete rotation means 360 degree, so it moves goes 47 centimeter in 360 degree, then 1 centimeter will be equal to 360 by 47, that is around 8 degree. So 1 centimeter, it is equivalent to 8 degrees. So if I want to give the second stimulation at a distance of 1 centimeter, so what should be the angle between the two striker? That should be 8 degree. Now come to our case. Here in this experiment, we have seen that the total duration of the latent phase is 1 centimeter. So first half is 0.5 and second half is 0.5. Then what should be the gap between two striker if I want to give the second stimulus at the latent phase? That is very easy. We know that 1 centimeter, it is equivalent to 8 degree. Okay, so if I want to put it in the 4 centimeter, it should be 4 degree. Okay, so we know the striker should be separated at 8 degree to get a distance of 1 centimeter between the 2 points of stimulus on the graph. So to get a second stimulus in the first half of the latent period, that is within 0.4 centimeter, the angle between the strikers should be less than 4 degree. So the striker should be separated at more than 4 degree or less than 8 degree. So in this way, 8 degree is equal to 1 centimeter, and we know that if we want to give it in the less than 0.5 centimeter, so it should be more than 4 degree but less than 8 degree. So in this way we can calculate the distance between the strikers and to see what happens in different two successive stimuli at different setup. Okay, so this is the graph we get when we give two successive stimulus. In the first we stimulus, it is a SMT. Second stimulus, it is applied on the first half of the latent period, so there is no change. Second stimulus S2 in the third picture, during the second half of the latent phase, there is again no change because the nerve is in the refractory period. Then in the next phase we have given it in the phase of contraction. So we can see that obvious change, there is summation of the activity and rise in height of contraction. And then the last one, we have given the second stimulation at the relaxation phase. We can see that the curve has not reached the baseline. Before reaching baseline, at the point of stimulus, it is again gone up and the height of contraction was more. So in this way we can adjust the different position of the graph. So this is the calculation and this is the graph. Now come to the another important part, that is a drum. It is 6 inches into 6 inches cylinder with a white glazed paper pasted on it. A drum grip latch provided on the top of the drum fixes the drum to the axle and prevents the drum from sliding around the shaft. It is smoked with kerosene soot, and while using ink pen writer, glazed paper without smoking is used. Okay, so normally we get the all these simple muscle twitch graph, we can see that it is on the black paper, but here we can see that it is surrounded by a glazed paper. So how it is possible? Actually we do the smoking of this curve. Okay, the drum pasted on the pasted with a white glazed paper, it is placed on a horizontal arm of a smoking stand, and the kerosene flame is kept below the drum and it is smoked uniformly by rotating it manually. So in this way we actually do the smoking of the drum, and on this smoked drum we actually take the uh, recording. Okay, so this is the smoking of the drum, you can see here. So after smoking of the drum, we take the recording and then we do the varnishing. It is a process by which the graph obtained on a smoking paper is fixed, made permanent. After the event recorded on the smoke paper, the time tracing is taken with the help of vibrating tuning fork and the graph is duly labeled. Now the paper is released from the drum by cutting it from the joint position. It is then dipped in the varnish tray. It contains 2% solution of resin and methylated spirit. The paper is then hung with a certain time to make it dry. And note that pollution concern has led to use of ink pen marker on the glazed paper. So after varnishing we get this. So in the previous slide you have seen that that varnishing is done with the help of 2% solution of resin and methylated spirit, and now when we have taken the recording, we have leveled it, we have taken the time tracing, then we will separate it, we take it out and we will make it permanent by dipping it into this solution, and you will dry it and make it permanent. So after that we will get this permanent curve. This is the curve of SMT that you see in the your classroom. Thank you.